Thank you so much, Angela. Welcome and thank you for listening. So I was really quite thrilled to be able to talk about protein crystallization because it's such an interesting topic. And with all due respect to the people who do just straight crystallography, crystallization is really the last interesting part of crystallography. Everything else has been solved, maybe. All right. So what I was going to do was just, first of all, introduce what I'm going to talk about in this particular seminar. And it's going to be sort of broken down into four major parts. The first is just an introduction, and then going to talk a little bit about what you need to know before you start moving into a protein crystallization strategy. Or not necessarily what you need to know, but what you should try to find out anyway. And then we'll move into the two different parts of crystallization, talking about first screening, and then we'll talk about optimization once we've moved through that screening strategy. And I'm still not quite used to this presentation mode, so if I stumble, if I fall, if I start making rude noises and loud swearing, that's all my problem and does not reflect at all on Rigaku. That's just me. Okay, so what is protein crystallization? What we're trying to do is we're trying to coax our protein to come into a nice ordered array so that we can shoot X-rays through it so we can get some information from it. And how we generally do that experiment is we take a purified sample of protein, we mix it with one or probably lots more of different stuff. And that stuff is often referred to as a reservoir condition, a well condition, a crystalline, a cocktail, but it's just stuff. 2 to 5 different chemicals at different concentrations. And then these are set up in various ways. They can be set up either in a hanging drop or a sitting drop vapor diffusion experiment where we mix the crystalline with the protein and set that up against a reservoir so that we can have some equilibration against that larger puddle of the stuff. Or we can set it up in a microbatch or a batch experiment where we mix the two things together and we just wait. But that's actually the common thing about all the crystallization experiments is that we wait. And so this blue arrow down here actually is a really condensed version of time. And then hopefully we get big, luscious crystals at the end. And if anybody is impressed by my blue crystals, you can do this at home too. That's blue food coloring from the supermarket mixed with lysozyme. All right, so moving on. And then, of course, one can think about crystallization experiments in a little bit more of a sort of theoretical way, and the way we usually think about these is through the phase diagram. And we move between different phases where we go from undersaturated, where the protein will never come out of solution, to oversaturated or supersaturated, where the protein will come out of solution, but not necessarily its crystalline form. And there's different paths that you can take through the different phase spaces which are available to you. I'm not really going to talk about this too much, but it is a nice thing to think about. The problem with the idea of a phase diagram is that to understand a phase diagram, you have to do an awful lot of work to get that phase diagram set up in the first place. And we usually run out of protein way before we even start mapping that out. So phase diagrams are pretty well mapped out for lysozyme against sodium chloride, but all other systems that are still just open saliva. Question that might arise is, well, why do you think that I was asked to give the seminar? Well, we have a little bit of background here which might, I don't know, justify the choice that Angela made here. I am associated with something called the Collaborative Crystallization Centre, which is a service-based crystallization facility in Melbourne, which is in Australia. And then I was going to take a little bit of time out to explain where Australia is. So I heard Martin Caffrey give a lovely answer to a question once. Somebody asked him, where's Limerick, which was the university in which he was residing at the time. And he pointed out that when people ask where Limerick is, they're usually meaning, where's Ireland? And I thought that that was a great thing to start with. So, where's Australia? Australia is this island in the southern part of the world which separates the Indian Ocean from the Pacific Ocean. Melbourne is the second largest city in Australia, and it is situated down here on the southern tip of Australia, sort of pointing towards Tasmania, so we could wave towards Tasmania. It's got a number of features, Melbourne. It has a nice river going through it. It has the Collaborative Crystallization Centre with some Rigaku stuff, in it, which you can see up in the top right-hand corner. It has the Australian synchrotron, most importantly, which is fabulous, and has changed our life down here in Australia. It is the host of the Australian Open Tennis Championship. And for those in the Commonwealth or the pink bits of the world, Melbourne hosts the Melbourne Cricket Ground, which is potentially the most famous edifice in the world if you're a cricket person. Now, the interesting thing for a crystallographer is that the Australian synchrotron is a round building which would fit just perfectly in the round bit in the middle of the MCG. I don't know if that was symmetric. All right, moving along. So what does the C3 do now that we know where it is down in Melbourne, Australia? We provide both screening and optimization services. So that makes us a little different from many other services which just do screening. We have about 150 different clients. They're both external to CSIRO, which is the Commonwealth, it is the government research organization, but we also have a lot of clients from research organizations and universities in Australia and overseas. We've been running for about 4.5 years now. We've set up close to 10,000 plates, which is about 1.5 million drops. About 18 million images have been collected of those drops, and the equipment that we use to do this consists of a Phoenix dispenser and mosquito dispenser, both of which we use sometimes in combination. We have a TCAN for mixing up stocks. We have 2 incubators, which are from Rigaku, and we have 2 imagers, which are also from Rigaku. And we have a bunch of secondary equipment, which makes that primary equipment useful. And the secondary equipment is, stuff that you'd normally see in a crystallization lab: microscope, sealers, lots and lots of labelers. Because the one thing that we've learned after 4.5 years of dealing with 150 people is that you have to be seriously well organized. And what I wanted to point out here is that of all the equipment that we have, the thing that we probably learn the most from is the images, because they allow us to capture information which we wouldn't otherwise see. And here's an example of that. So here is an example where we had glucose isomerase set up in condition A12 of the JCSG, and that condition is just a PEG condition with a little bit of nitrate in it. And it turns out that this is the time course of it. So that was day 1. This is day 1. Then this is day 3. We have these stunning crystals in this sort of a Stonehenge arrangement. Day 5, they've disappeared, and you can still see the shadow of them. But without that imager, we would never have necessarily seen those crystals. And then we actually have to step through quite a lot of the time course until we get to about day 50, until we actually see that more crystals do eventually grow, but they don't look anything like those really quite stunning radial crystals that we saw before. So that just shows you that you can find things when you actually capture the time course that you wouldn't have otherwise seen, and we find that really very, very interesting. And then if we move on, given that we find we have the equipment, we have the background, one of the things that I wanted to point out, and one of the things we've learned, is that crystallization is generally not interesting. In fact, I would say of my 150 users, about none of them are interested in crystallization. What they're interested in is crystals. And then actually, if you point that even harder, they're not actually interested in crystals at all. What they're interested in is the biological information that you get from shooting X-rays through crystals. And so one of the things that we always have to be aware of is that crystallization isn't an endpoint. Crystallization is a sort of irritating necessity on the path to an endpoint. And that also then brings up this idea of the relevance of the information that you're getting. The relevance of the information will depend on the time in which you can get that information, but also on the resolution of the information that you get. If you're looking to do a fragment-based screening protocol and you're working with 3.5 angstrom system, that's not going to help you because you just don't have the resolution to give you the information. So you need to know going into a crystallization strategy what your limits are on resolution and what they are in time. Because if you can't work to that, you might as well not even start. And certainly not waste your time doing it. when we get started, we have to appreciate, and I'm sure you all do, that all proteins are different, they behave differently, and the thing that to really remember is that your crystallization strategies will change depending on the protein that you have. One thing, though, is completely invariant in all crystallization strategies. If you go in with an expectation that nothing will work, you won't be disappointed. And so I show here a picture of some chap who's just had it pointed out to him that his crystallization experiment did not work. So to avoid things like that, we just walk in with an unrealistically low expectation. So expectation management, keep that in mind. It's probably the most important thing a crystallizer can have. So when we start a crystallization experiment, we've either spent time making protein ourselves, or we have got that protein from somebody else who has spent a lot of time. The important thing is to get as much information about that protein into your headspace, from either yourself, from your purification, or from the person who did it. And these are a list of questions that I thought of really quickly. There's probably more you can think of. Let's step through some of these in a little bit more detail. How much protein is available? This is certainly a really relevant question. Crystallization is easier the more sample you have. I would say if somebody comes to me and says, hey, I'm interested in getting a crystal structure, if they don't have about 0.5 ml of concentrated protein, it's just going to be hard. Because with 500 microliters of protein, you can sit there and you can actually start thinking about doing different things. You're not paranoid the whole time that every single drop that you set up has to be absolutely exquisitely the right experiment, because otherwise you've just wasted the protein. You've got a little bit of flexibility, and that means that you do things. When you're super paranoid about every single drop being the right experiment, you tend not to do the experiments because they're too hard to make sure that you get exactly the right one. When you've got a little bit more security because you have more protein, you just do the work. It's much easier. The nice thing about 500 microliters of protein that even if you're setting up by hand or if you're setting up by machine, it gives you about 20 plates. And 20 plates, you can get a long way in 20 plates, assuming that either a 24 well plate by hand or a 96 plate by machine takes about 25 microliters of protein. And from my previous life at SGX Pharmaceuticals and from my work here, we have found that the best correlation between crystallizability and any other factor seems to be expression levels. If you have a protein that expresses seriously well and purifies really easily, then it is more likely to crystallize than the problem children that you have during the expression and protein purification stage, which makes sense because they're both a function of the stability of the protein. Next big thing is to know how concentrated your protein is. Crystallization is a precipitation process. So if your protein is not high enough concentration, it will never come out of solution when pickled with the crystallization solution. And so before you go and set up 10 plates with your protein, check to see that it is in an appropriate concentration range. Proteins can vary enormously in their solubility, and crystals have been obtained with protein solutions which range from 0.2 mg per mil to 200 mg per mil. So, the idea of saying a protein should be at 10 mg per mil is, yeah, a bit of a hand-waving thing. Try an experiment first. Set up a single plate and see if you get about 50% of the drops precipitated, or do a pre-crystallization test. A pre-crystallization test is when you have a couple of solutions which are indicative of crystallization space, usually a 2 molar ammonium sulfate solution and a 30% PEG 4000 or 3350 condition. Set them up in a single experiment or two experiments, one with the ammonium sulfate, one with the PEG, in the system that you'll be using, and then check the results. If they're both heavily precipitated, you need to back off your protein concentration. If they're both clear, you need to push your protein concentration up. You can do that either by spin concentration, dialysis potentially, or by changing the ratio of the drop when you're actually doing your screening experiment. You do 2 parts of protein to 1 part of crystal, and you will end up with a more concentrated protein in the end equilibrated drop. the buffer that the protein comes in is enormously important to the success of any crystallization trial. Particularly if you're working with somebody who is also working with an NMR spectroscopist, you need to stamp your feet firmly, actually, and say, no, you don't really want it in a phosphate buffer. Phosphate buffers are great. Proteins like them. NMR spectroscopists adore them. However, they've got a problem in that they will give you crystals in crystallization screening. They won't necessarily give you protein crystals. Magnesium phosphate is phenomenally insoluble, as is calcium phosphate. If you look here, there's a table of different phosphate salts and the solubility product of those different salts. And all of these really negative exponential numbers mean really, really insoluble. Compare that to sodium chloride down the bottom, which is actually very soluble, and you can see that this is why we get so many phosphate salt crystals during a crystallization experiment. So try to stay away from phosphate. Somewhat similar with citrates, somewhat similar with some of the other protein-happy buffers. The other thing is to remember is that you can make your protein in high salt buffers, and that's great, except that means that your protein crystallization experiment will tend not to perturb that system as much as it would if your protein were in a more minimal buffer. So that's also something to remember. And a further thing there is if your protein only stays in solution in 10% glycerol and 500 millimolar sodium chloride, you don't have soluble protein. What you have is you have protein which is being shocked into suspension by all the other stuff in that solution. So one sort of hint is if you need to have a very concentrated buffer to keep your protein in solution, you need to, even before you start crystallization, go back and work on your protein to make that a happier and more stable protein. If you get your protein from a third party, you really need to get the details of the buffer. A lot of people in CSIRO use TBSA, and I had no idea what TBSA was until I actually got somebody to describe it to me. It's tris buffered saline with azide in it, okay? Initials don't necessarily mean the same thing to you as they might mean to somebody else, so really check that out. Other things important to know, the sequence of the protein is important to know. Sometimes people sneak large tags onto the protein without telling you. A maltose binding protein tag can really affect your protein crystallization, as can just a his tag. So it's really good to know the sequence. If nothing else, that allows you to estimate the molecular weight. It allows you to estimate the extinction coefficient. You can start doing some playing with some of the bioinformatics, which is available to find out whether your protein will crystallize. And I give it three of the more common protein crystallization predictors are listed here. In all honesty, I don't think any of them are particularly strong at the moment, but the more we use them potentially, the more they will get better. But the sequences are very important. We are interested in having happy protein or stable protein, that's the picture of horses. And the way to do that is to make sure that everything that the protein needs is in the solution with it. So that means if it has intrinsic metals, make sure that they're in there. Make sure that any cofactor is associated with the protein. Substrates, not so good generally to put in with your protein, because if your protein is active, it will turn over and you won't have substrate anymore. Inhibitors tend to be good because they do tend to lock down your protein into a single conformation. And then another thing to ask about is the idea of batch-to-batch variation. Crystallization can be particularly sensitive to batch-to-batch variation, even if your activity assays and your protein gels don't show it. And you only tend to realize that this is a problem when the second batch of protein, which you swear blind was produced in exactly the same way as your first batch, doesn't behave the same way. And a particularly egregious example of this was when I was in Uppsala. We were working on rubisco, which is the most abundant enzyme in the world, and this particular rubisco sample was from spinach. We got crystals which grew to ridiculously large millimeter sizes practically overnight. If the spinach was grown in spring, if the spinach were grown in autumn, it didn't crystallize at all. And Goodness knows why. Still don't know. Maybe it was a phase of the moon. Don't know. But the batch-to-batch variation was quite stunning. And so that is something to be aware of the whole time. This also suggests that it is better to do one large prep of protein and then work out how to store your protein than many smaller preps, because you minimize that batch-to-batch variation. Moving on to how can your protein be stored. It's so important because crystallization is this enormously time-dependent event. So you can't just set up an experiment and expect to see a result. You set up an experiment and it might take days or weeks or months, some people years, I'm not that patient, for your experiment to actually produce results. And if you want to then follow up on the results from your initial screening, and the initial screening takes months, how are you storing your sample while that process is going on? And there is an apocryphal story about frogs. I don't know if it's true or not, but the idea is that if you take a pot of water and you put a frog in it, and then you very, very slowly heat it up, the frog never quite works out when the water gets too hot for comfort and then boils to death. However, if you have a pot of boiling water and put a frog in it, it jumps out immediately because he knows it's too hot. And so if you have your protein, which is sitting in an ice bucket in the cold room, and you religiously change the ice so it doesn't just float, that's great. When is that protein likely to be bad? You can sort of say, you know that in 10 years' time it will be bad, but is it bad in a week's time? Is it bad in 2 months' time? Is it bad in 10 years' time? If you can work out how to store your protein frozen, that gets rid of that whole frog, when is it bad issue. We try to encourage people to store their proteins in aliquots, which are 50 to 100 microliters, which have been snap frozen in liquid nitrogen in PCR tubes. And that's just to ensure that you get the fastest freezing possible. And then when you come to do your experiments, you use frozen protein both for screening and for optimization. So you're always working with the same system. When you freeze, you will lose some activity, but not generally as much as you'd expect, usually about 5 to 10%. And then the other things you can know about your protein. How pure is it? Yes, purer is better, but we have to remember that protein crystallization is a purification event. Is your protein glycosylated? Generally, less glycosylation is better because it's just stuff, flatten and breathe. Is it aggregated? Generally, once again, less is better. Most people look at these things as post-failure rationalizations because it's easy to get hung up on purity. It's easy enough to get hung up on all these. Do the experiment first, but be aware. This particular gel I'm showing here is a gel of the samples that came in recently into C3. We run a protein gel of all of our samples. We put these up on the web for everyone to see. However, we only identify them by sample ID, not by the person who made them. Now, I would look at these and I would say there are some lanes of protein here that I would not at all be surprised if they don't work too well. Lanes 1 and 2 are probably very similar proteins. They're not very clean. Lane 4, however, that looks like a very appropriate sample for crystallization. Lane 6, somebody is living in La La Land. All right. But even so, get concerned about purity. Don't get too concerned about purity in the first instance. So other things that control besides just your protein, you need to control the other parts of the experiment. How old are your screens? How old are your stocks? How well are they made? And you need to also control your consumables, your pipettes. Are they actually pipetting and dispensing accurately? Are your seals fresh enough that the adhesive still works? Do you store your plates in a dust-free environment? The really big one, though, is to manage your data. Yeah. If you have any sort of automation at all, it is extraordinarily easy to set up more plates and you can comfortably capture the information with a pen and a piece of paper. We at C3 probably spend somewhere around a third of our time just managing the information flow into and out of the center. So it's really important to realize that if you can't find the information about a particular experiment, you may as well not set up that experiment because you've just wasted time. So it's always a question of being able to go back and find out what you did. Moving into some more examples, one of the things that we do here is we make sure that our all of our chemical stocks are made consistently, and not only that, but they're labeled consistently. So if you can see on the right-hand side here, we label things with their concentration, with their chemical, with the date, who made them, that they're stored appropriately, and the image of different colors on the left-hand side is showing this. This is an image of different PEG solutions which were made up in the C3 lab. And the color comes from putting a universal dye in, and that's shown down the bottom as a pH gradient going from pH 4 to pH 10. And so you can see pink means that the product is acid, and blue or purple means that the product is basic. You can see that all the PEG stocks that we had in C3, except for PEG 3350, were really quite acid. And the one that we were interested in was this PEG 2000, which was sitting here down, I don't know if I can actually try to highlight here, which was sitting here, there you go, beautiful, which was sitting here down here, and we're going, wow, that is really quite acid. Then we wondered whether it was the stock that had gone acid or whether it was actually the PEG solid that went acid. So we used the same solid, and we made up another solution, and we found out that that was actually really quite neutral. And this pink one, the acid one, was only about 6 months old. So ever since then, we have now had a policy that we keep our PEG solutions in the dark, in the cold, and we throw them out after 6 months, because we want to make sure that we have some consistency in how things behave. And similarly, we do the same thing with our stocks, not just with the chemicals. So when you actually buy your screens, you often buy them in tubes and then dispense them into depot blocks yourself. We actually weigh our screen blocks when we do this. And so here's an example of the packed screen, which we got in tubes, we dispensed into blocks, and that block weighed 280.24 grams when we set it up. When we come to use that block in N months' time, we can actually just shove it on the scale again and make sure it weighs the same. If it weighs the same, it's probably good to use. If it doesn't weigh the same, we throw it out. We don't even bother thinking about where it might have gone wrong. The other thing we check is that using that same pH assay, we can actually see whether the screen has changed over time. And this actually starts really explaining why you mightn't get consistent results between your screens and your optimizations, because the pHs do change over time. Here's an example of a color pH plate of the Hampton HT crystal screen. So you can see that there is quite a lot of information in the pH. This graph here is actually interesting. What we had is we had two versions of the nucleic screen, and one which had been stored well and one which had been stored badly. And we did the pH assay like this on both the old and the new nucleic, and then we compared the pH, and you can see that there's up to 2.5 pH unit difference between the two screens when one got old. So that's an incredibly important thing to remember, that not just your protein, but the other parts of your experiment need to be controlled as well. And then the data stuff, well, how do we manage data? We have to be particularly careful because we are dealing with 150 different users. What we did is we borrowed an idea from James Holton, which is from a synchrotron. Synchrotron has this same idea. They generate a lot of data, but it's not really their data. And they manage that by burning DVDs, and we do the same thing. So that we have a database where you can actually see the information about the experiments as they're set up. And this information is available that you can come in, look at your images, see what was in the experiment at the time, and then annotate it as you want. But after the inspection cycle has finished, the images get burnt to a DVD along with the chemical information, and we present a viewer, which means that you can get to that information very trivially. And then we burn 2 copies of this, 1 copy that we keep, 1 copy which we give to our users, so that the users then are the primary holders of their data. So that is sort of what you need to have set up before you even start doing the experiments. And then you move into the actual crystallization experiment. And the first thing that you generally do is you screen. And I think it's very, very important to remember that the goal of screening is not to get a well-diffracting crystal. If you do, that's a bonus, but that's not what the goal is. The goal of screening is to find a starting point for optimization. So a good outcome from a screening experiment is to find out what temperature the the protein prefers, what is the suitable concentration to try, some knowledge about the protein's preferences in terms of precipitants, whether it prefers, salts versus pegs. If you can get some initial crystals to use as seeds, that would be fabulous. A list of things which seem to positively affect crystallization. So if you've got something that says, my protein really does prefer to be at 20 degrees, it likes higher salt and it prefers low pH, that's a really good start out of your screening. And screening is actually very easy to overdo because there's a lot of tools out there to make it easy. There are over 220 commercial screens available from about 7 different vendors. Most of these are initial screens. And so you could sit there and just screen all day. There's a couple of things which will limit how much you screen. One is that the screens aren't particularly cheap. Another is that you would have to find a place to store all of those 14,000 odd conditions. Another thing to realize is that there is an awful lot of duplication in the screen. So, but every novel condition in that set of commercially available screens, there's probably about another 3.5 conditions which look about the same. We have built a web tool that can help with this, and I have a movie, I don't know if it'll work, but I can discuss that later. What the C6 does is it has a distance metric which allows you to map differences between different screens and see which screens are the same. And that's quite hard to do by eye because the screens don't necessarily have the same chemicals or have the chemicals described in the same way, in the same order, in the same drop positions, but they are, beneath that, are the same. So the other thing is, as I keep saying, don't over-screen. You are going to need some protein for optimization. And it's always interesting when you're screening, are you interested in screening diversity, which is different screening conditions, or are you interested in testing duplication? Crystallization is intrinsically a stochastic, a random event, because nucleation, which is required for that crystal to grow, is a random event. And so sometimes you may get better results by actually setting up the same drop lots of times rather than setting up lots and lots of different chemical conditions. So you always need to balance that need between duplication and diversity. And I think at the recent ICCBM meeting in Dublin, there was quite a lot of people who came out and said, we find it more important to actually test different ratios of our protein against a smaller set of screens. And so we will halve the number of screens and double the number of drop ratios we try, and we get better results. The strategy that you use for your screening is going to depend on how much protein you have and lots of things. However, if pushed, then this is what I suggest. We have a structural genomics initiative here at CSIRO, and the screening strategy that we have set up for these people, and they tend to get quite a lot of protein, so we're not protein limited. We set up the JCSG, which is a good sparse matrix screen. We set that up at 2 different temperatures, and that's testing diversity and duplication and temperature by setting up those 2 plates. We then set up packed, and that gives you a more tighter view into the pH side of crystallization space. But the beauty of the packed screen is it's very systematic. So you can actually start pulling information about the pH behavior of your protein, particularly from that screen. And then the third screen that we set up is something which is called the PS gradient, which is a screen that we developed in-house, and it's just a very simple ammonium sulfate versus pH and a sodium malonate versus pH grid on 96 blocks. And with those 4 screens, we cover both the PEG side, the salt side, the duplication, and the diversity. And that seems to work really quite well. And then here is a picture of the C6 web tool, which I encourage you to use. You need a username and password to use it. You can come in as guest at C3, and the password, hopefully easy to remember, is Vegemite. And here's a screenshot of one of the Java viewers that you can get out of that, and I really don't think my movie is going to work. But here is where we decided to say, okay, what screens do we have which are available to the Crystal Screen HT, which is the Hampton product? And you can see that then around there you can see some of the other screens which are similar. So the basic kit from Sigma, the structure screen 1 from Molecular Dimensions, the classic suite from Hyogen, and the JBS Basic HTS screen are essentially the same screen. Crystal Screen 2 here is another Hansen product, and it is completely contained within Crystal Screen HT, so that's why it's green. It means that it's a subset of the screen that we're looking at. So screening, second round, sometimes you might get microcrystals or other things, other solids, which aren't suitable for X-ray analysis, but they can be used to help you in your screening. So there is a process where you squash them up, make microseeds, and then go back and screen, but this time use those seeds from the first round. This tends to be a really useful technique, and you can pull out crystals which you wouldn't have otherwise found in your screening. You don't have to use different screens. You can go back into the same screens that you used for the first round, because this time what you're trying to do is jumpstart yourself over the nucleation step. The issue with this, even though it's a very powerful technique, is you will tend to grow lots of salt crystals this way, because you're starting to mix potentially incompatible crystallization cocktails together. And I show here an example of 3 different salt crystals, all nice salt crystals, all actually obtained through matrix seeding. And so then from screening, which is actually pretty simple, and that's why people like it, because you don't have to think that much about it. We move to optimization. The first thing is to get used to the idea. You will probably have to optimize to get well-diffracted crystals. But there is a basic flaw with optimization, as she has done at the moment. We tend to optimize for visual perfection. Humans like symmetry. We like crystals. So we look for the biggest, most optically perfect crystal and say, that must be it. That's the one I'm following. But what we're actually interested in is diffraction quality. And unfortunately, visual quality and diffraction quality aren't particularly well correlated. And in fact, the 2 pictures I show here are trying to demonstrate that. The picture on the left looked like a sort of aeroplane-shaped blob after the wings had blown up because the jet engine lost bits. All right? That's being diffracted to beyond 3 angstrom. The gorgeous gems on the right-hand side didn't give a spot, not a single. I'm still bitter about the fact that they didn't give a single spot. So the thing that you need to think about in your optimization strategies is what is a hit? How many hits do you have? And hits come in all shapes and forms. Clearly, if you get a nice big crystal, yeah, that's a hit. But, you know, a sandy type microcrystalline precipitate is certainly a hit. And all of these images here are showing very good starting points for optimization. And in fact, some of these on a microfocus beam mine might actually be the holy grail of getting something suitable for shooting straight out of a screen. So what are the things that you can do in optimization? They basically, you can do anything you like. There's an infinite number of things that you can do, but they come down to 4 different approaches. You can fine screen, which is just tweak the chemical conditions which you've already identified. You can seed, as we did in screening, but this time for optimization. We can start adding things to the conditions which we think are likely, and that would be additive screening, or we can start changing the kinetics of the experiment by altering the rate at which things equilibrate. So in fine screening, what we tend to do is we say, okay, I have identified these factors are important. I will step through the factors that I think are important, generally in a grid. Grids were done initially because they're easy to dispense. Yeah, so you can do a squirt of chemical 1 in well 1, 2 squirts of chemical 1 in well 2, and so on. And they work fine, but they are sort of limited in how many things you can test at once. Random combinations can be done with automation, because you can start getting machines to do this mixing. If you try to make up a random set of 96 conditions using 15 different chemicals by hand, there is a really good chance you're not going to end up with what you think you had. But if you have a TCAN or some other device to do that, then this works really well. And in fact, Brent Segelke in 2001, I think it was 2001, had developed a program called Webtool, which used this sort of random approach. And it's a very powerful way of mixing the factors from more than one hit into a fine screen. And so here I'm trying to actually display why your grid screens may be appropriate. So say you get a hit at this position here, and you do a grid screen, and you test 3 factors, and you can actually quite nicely miss your optimal, which is here. If you're doing more of a random walk around there, there's much more chance that you will actually start heading in the right direction because you can see where to go. So one of the things that people have found, because with the advent of automation, people tend to screen in 96-well plates and then start moving into 24-well plates for optimization, and potentially even moving from sitting drops for the initial screening to hanging drops for optimization. And often it just doesn't work very well, even though you think everything is well controlled. And the things that may change, you may have different stocks, you have different mixing, you have different drop ratios, different surface materials, different surface area to volume ratios, different kinetics of equilibrium. You might have different vapors outgassing from your grease or from your adhesive or from your tape. So you might want to think that just one way of making this whole crystallization process more robust is to optimize in the same solution, in the same system as you use for screening. So if you can optimize in a 96-well plate and screen in a 96-well plate, that actually probably helps you. When we were at SGX, we were actually doing hanging drops in 96-well plates and optimization in 24-well plates, but we would often find that we'd have to go through a 48-well hanging drop plate intermediate to make that process work. So moving away from fine screening into seeding for optimization, there's sort of 2 aspects of seeding. One is to enhance the reliability of your crystal formation, and that's particularly useful when you're in the sort of production mode of crystallization. And the other is to improve the morphology of your crystals, because you can move your crystallization experiment away from being very supersaturated into just the supersaturated region. A couple of examples. So we had a project where we were trying to grow several thousand Trypsin crystals for a particular sort of high throughput approach to fragment screening. And we found a nice optimized crystal condition for growing Trypsin crystals, which, quite honestly isn't that hard to grow. And here are 96 drops, all set up with exactly the same condition about 24 hours after we set them up. And what you should see is 96 gray blobs which have no color around them. Then if you actually look at this next slide, this was the second subwell in the same place, but the only difference between the top drops, or the drops in the first set of subwells, and the drops that I'm showing here now is that these ones were seeded. And the colors around the wells indicate that somebody has looked at that and said that there is a crystal that's worth harvesting in there. And you can see here that we've got about 70% of the drops which were seeded, which were identical in composition to the drops which weren't seeded, have crystals. And that was just the addition of nucleation. So that's increasing our reliability. And then this is an example from Therese Bettiforsch, which is showing how you can improve the quality of your crystals by going through rounds of sequential, sometimes seeding. So she started with these furry things, which clearly crystalline, but clearly not going to be very useful in the beam, moved through this intermediate, which are probably useful now, but weren't useful with the X-ray technology which was available then, to this honker, which diffracted diffracted X-rays from here to Sydney and managed to solve one PhD student's life problems. Additive screens. Additive screens we use a lot in C3. We find them phenomenally useful. We have the Hampton additive screen, we have the OptiSalt additive screen, we use a detergent screen, we use silver bullets, and we have an ionic liquid additive screen, which we use. They're all good. They're all good. But if I had to just choose one, I would probably go for the Hampton additive screen. And the idea here is that you have a base condition which shows something promising, and then you just start tweaking it by adding something else. And so here's an example from the C3 where this was the best thing that came out of the initial screening. And then with the addition of additive screening, and this was the Hampton additive screen, we started getting some much more interesting results. And this is from the same screen until, one of the conditions in the additive screen showed that, which was then suitable for X-ray diffraction analysis. And then the fourth aspect of crystallization, potentially the one that isn't used so much, is kinetic manipulation. And this is the thing that kills you when you try to move from 96 wall plates to 24 wall plates, because it really does affect the experiment you do. But you can do that deliberately. You can have different drop sizes. Larger drops will tend to equilibrate more slowly and will give you different results. If you use different drop ratios, you can change the equilibration rate and you change the endpoint of that particular experiment. Different temperatures, of course, are not just affecting the temperature, but they're also affecting the rate at which things equilibrate. You can set up droplets over alternative reservoirs. So if you set up things over salt rather than over the actual reservoir condition, that can profoundly affect the behavior of that particular experiment. And that turns out to be quite a cheap way of actually extending the screening as well. And the picture that I'm showing here is actually Falmerton set up, Falmerton pre-mixed with a little bit of tartrate and some ADA, and then set up over a gradient of sodium chloride that went from 0.7 molar sodium chloride over here to 2.5 molar sodium chloride over here. But the drops were identical and didn't contain any of the reservoir in there at all. And actually you can see that across this gradient, as you move across here, that you go from being undersaturated, beautiful crystal, and then you can see this is a really good indication that the phase space is not smooth, because we get a crystal, then we get no crystals, we go back to crystals, microcrystals, back to large crystals, and then microcrystals again. And so it's a really nice indication that phase space isn't useful, but this alternative reservoir gradient screening is a really very, very nice way of moving between sort of microcrystalline and single crystals. Another way of doing that, of course, is adding oil over the reservoir solution, which changes the rate of equilibration. And then, of course, just changing from sitting to hanging drops can do the same thing. So what we need to think about is working backwards. We know from our own experience, we're not actually interested in crystals. We're interested in the biological information that comes from the structure, but we're interested in doing that in the most efficient way. And you could always squander time in this process in so many different ways. You know, one of the first things to do is to believe that real men build into crappy maps. No, real men go and get better data so that the maps aren't crappy. That will tend to speed things up. Yeah, processing lousy data, once again, that's what heroes do. No, 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 no. It's much better. I mean, you can always either try to get better crystals, or with the crystals you have, get better data. Talk to your beamline scientists. They're really good at getting the best data from the crystals at hand. If you're growing lousy crystals, grow better crystals, get better protein. And if you're fighting lousy protein, get a better construct, get a better expression system, purify things differently. What we have to remember is that everything we do comes down to the protein. And with that, I'd like to stop to say thank you to the people who work in C3 with me. That's Christine, Shane, Tam. Tom allows me to work in C3, and Vincent is our software engineer who is fabulous. Thank you so much to Angela for allowing me to speak, and thank you all for listening.